Thank you for the introduction, and I really thank the committee of KSNCB and Merck for giving me such a great honor and chance to present my work in front of you. So the topic that I'm going to present today is metabolic and enzyme engineering strategies for the production of carminic acid, which is a natural red colorant. Sorry for the delay. So carminic acid is a natural red colorant that is widely used for food, cosmetics, and pharmaceuticals, and it is classified as an aromatic polyketide. And as you can see from this photo, it is also famous for being used in lipsticks and strawberry milk. However, what many people do not know is that it is actually being extracted from bugs, the cochineals. However, this has several problems because the cochineals can only be grown in a limited area, and also the production process is highly inefficient, as about 70,000 cochineals are needed to produce 0.4 kilogram of carminic acid. And the extraction process is highly laborious. And moreover, the insect protein contamination included in the product might cause allergic reactions in people. However, as it is still one of the most stable red colorant produced from nature, the need of an insect-free method of producing carminic acid has been increasing. So before going into the detail, let me briefly explain about polyketides. So polyketides are complex natural products that are produced by repetitive clasing condensations of small carbon units such as malonyl-CoA or acetyl-CoA. And the core enzyme groups that are required for the production of polyketides are called polyketide synthases, or PKSs in short. And there are three types of PKSs according to their biosynthetic mechanisms, type 1, 2, and 3. And in this research, we focused on type 2 and 3 PKSs as they can produce aromatic polyketides, and because carminic acid is an aromatic polyketide. So this is the carminic acid biosynthetic pathway, and the first step, as you can see here, it is a polyketide synthase. And by using the type 2 and type 3 PKSs adopted from Photoropsis and aloe, we first checked whether these type 2 and 3 polyketide synthases can produce aromatic polyketides. And to make the long story short, when we combine and tested different components of the type 2 and 3 PKS constructs, we found that both of the type 2 and 3 PKSs could produce similar amounts of SEC4 and SEC4b, which are the shunt products for carminic acid biosynthesis. However, what was interesting is that when we introduced cyclases from Streptomyces for the production of flavochromistic acid, which is the precursor for carminic acid, we found that actually type 2 PKS resulted in significantly higher production of flavochromistic acid, which we thought that this is due to the better interaction of the cyclase with the type 2 PKS constructs. And in order to prevent melanin synthesis by flavochromistic acid, we added ascorbic acid, an antioxidant agent, and we could produce about 180 milligram per liter of flavochromistic acid from glucose. The real problem was start from here. So from flavochromistic acid to carminic acid, two enzymes are required, monooxygenase and C-glucosyltransferase. However, the first enzyme, monooxygenase, has not been reported. Therefore, we employed biochemical reaction analysis in order to elucidate the enzyme. So that means that we tested different candidate enzymes that can perform similar reactions. And by testing these enzymes, Sorry for the technical issue. We found that DNRF from Streptomyces could successfully produce carminic acid. And we also wanted to test the next enzyme, C-glucosyltransferase. In fact, the C-glucosyltransferase from the native producer cochineal has already been reported. However, no matter how we tried, we could not succeed in producing DC2, which is another precursor to carminic acid. So we concluded that this CGT from the cochineal was not functional in E. coli. In this regard, we employed the same strategy that we employed for elucidating monooxygenase. And we also performed biochemical reaction analysis. And by testing the 7 candidate enzymes, we found that only GT-CGT from Gentiana flora could produce DC2. Even though we found these 2 enzymes that are capable of producing carminic acid, we found that because the native substrate of these enzymes were not flavocarminic acid, the activities of these enzymes were too low. So there was a need of developing enzyme mutants with enhanced activities. However, one more problem is that the crystal structures of these enzymes were not reported. So we first performed homology modeling of DNRF using modeler. So using a template enzyme with a known crystal structure with high sequence identity with our target protein, DNRF, we performed homology modeling and generated a structure model of DNRF. Then we performed docking simulation by docking flavocarminic acid into the DNRF structure model using Autodock. To make the long story short, by testing these different enzyme mutants, we successfully enhanced the activity of DNRF about 2.5-fold. So if you look into the detail of simulation results, we can see that the binding energy of the mutant enzyme with our desired substrate is lower than that of the native enzyme. That means that the mutant enzyme is predicted to bind better with our desired substrate, flavocarminic acid. And on the other hand, if we compare the binding energy with the native ligand, aclavinone, we found that the predicted binding energy with the aclavinone was predicted to be higher in the mutant enzyme. That means that it is less stable. And for the GT-CGT, we also performed same strategies for developing enzyme mutants. So first we performed docking homology modeling, and then we performed docking simulations. So to make the long story short, we also succeeded in enhancing the activity of the C-glucosyltransferase by introducing 2 mutations and could successfully enhance the enzyme activity by 5-fold. And if you take a closer look into the simulation results, you can also see that the binding energy with the transition state analog of the flavocarminic acid was lower than that predicted from the native enzyme. That also means that the mutant enzyme can bind more stably with our desired substrate. And if you look into the active site at the figure below, you can see that the C2, where C-glucosylation takes place, is open when compared with that of the native enzyme. So by introducing these mutations, the C-glucosylation reaction can happen much easily. And because C-glucosylation is an important reaction for producing natural products with enhanced properties such as solubility, bioavailability, and stability, we thought maybe we can also apply this mutant enzyme for the production of other polyketides as well. So we chose aloesin, which is found in aloe plants and which is also used for cosmetic ingredients due to its anti-inflammatory and skin whitening properties. And if you look into the biosynthetic pathway of aloesin, you can see that it also requires a C-glucosyltransferase, which has not been reported yet. So after we introduced the mutant C-glucosyltransferase, we succeeded in producing aloesin from glucose for the first time. So we inferred that the mutant enzyme that we developed in our study can be a generally applicable C-glucosyltransferase for the production of aromatic polyketides. So finally, we combined all the strategies that we developed in our study and successfully produced carbonic acid from glucose. By performing fat-batch fermentation, we produced 0.6 milligram per liter of carbonic acid from glucose. And this result has been published in JACS this year. So to sum up, we first employed type 2 PKS and Ju-IJ cyclases for the production of carbonic acid. And by employing biochemical reaction analysis, we successfully elucidated a monoxygenase and C-glucosyltransferase for the production of carbonic acid. Next, we performed docking simulations to enhance the activities of these enzymes. And we found that the GT-CGT mutant is a generally applicable C-glucosyltransferase as exemplified by the alloysin production. And lastly, we combined the strategies and successfully produced carbonic acid from glucose. So thank you very much for your attention, and I would like to especially thank my advisor, Professor Sang-Yep Lee, and my colleagues, and I also thank the ministry for supporting my research. Thank you.
Congratulations, Senior. Thank you for your nice talk.